said everything gonna be alright. I said everything gonna be alright.
Right. Everything's gonna be alright. Good day wherever you're listening from, and welcome to Indoor Air Quality Radio, IAQ Radio. It's Friday, August 19th, 2016. This week is episode 427. My name is Radio Joe Hughes, and we're going to reach back into the archives for a show we did on April 10th of 2015 with Dr. Jack Gilbert of Argonne National Lab. We talked about the microbiome of the indoor environment, one of the early extensive shows we've done on microbiome. Well done, and looking forward to listening again. I'm on vacation this week. We'll be back live next week, so enjoy the show.
John Donne Products, where restoration and abatement contractors shop. Visit them at their website, jondon.com. That's jondon.com.
Clean Facts, the #1 information source for cleaning and restoration professionals. Check them out at cleanfactswithanx.com.
IAQ.net and Healthy Indoors Magazine, a free online digital magazine for industry professionals and consumers. Subscriptions are available at IAQ.net.
And Particles Plus. They are engineers and manufacturers of feature-rich particle counters, air quality monitoring instrumentation, and vacuum pump technology. ParticlesPlus.com. Count on us.
Please be sure to thank our sponsors for their support of IAQ Radio when you inquire about their services and products.
All right, Dr. Jack Gilbert, he earned his Ph.D. from Unilever and Nottingham University in the United Kingdom, 2002. His postdoctoral training was at Queen's University in Canada. He subsequently returned to the UK in 2005 to the Plymouth Marine Laboratory as a senior scientist until his move to the Argonne National Laboratory and the University of Chicago in 2010. Dr. Gilbert is the group leader for microbial ecology at the Argonne National Laboratory and associate professor in the Department of Ecology and Evolution at the Department of Surgery at the University of Chicago, associate director of the Institute of Genomic and Systems Biology, and a senior scientist at the Marine Biological Laboratory. I think we've got some intro music for Dr. Gilbert.
Bacteria, you are my friend. You've been there from the start and you'll stay till the end. My body houses your microbiome, but somehow you make it feel like home.
Cliff, I don't know where you find these, but it was good. Let's see if we've got Dr. Gilbert on the line.
Absolutely, yes. That's a very interesting song.
It's we were looking at it. It was kind of funny. It had like 800 views on YouTube, so Cliff was one of the few that have seen it so far. Anyway, it's great to have you with us. I know you're a busy guy, and we're really happy you could pull away and join us today. Tell us a little first about what Argonne National Laboratory is.
Oh, yeah, great. So Argonne National Lab is one of the Department of Energy's national facilities. So these are laboratories around the country which were set up to try and focus scientific research in America and bring the best and the brightest to tackle the real tough problems.
How many are there?
There are about 8 or 9 national laboratories around the country. Okay. Argonne is one of the oldest. It was set up really to deal with Enrico Fermi's nuclear reactor that he tried to build under the football pitch at University of Chicago. When the mayor had decided he'd had enough of having a nuclear reactor in the city, they moved it out to the suburbs.
Interesting. And how did you get your early interest in microbiology, or is that something that came later in life?
It actually came later. I started out as an entomologist, and then I became interested. I studied butterflies, and then I became interested in proteins and how proteins function. What I did for Unilever was looking for proteins in Antarctica that could be used to make ice cream taste smoother. So obviously very, very important research. And then I don't know, I kind of stumbled into it in around 2004, 2005. There was just a buzz being generated about the microbiome, and I decided to start exploring microbial ecology, the bacteria, the fungi, the viruses, the archaea, the single-celled life on this planet, and what role it played in our health, our ecosystem functions, and everything.
And you sound kind of excited about it now. Is this a new passion? I mean, is this something you think you'll stick with?
Oh, absolutely. I mean, it's a great time to be a microbial ecologist or a microbiologist. We have a lot of fun tools to play with. We have an unprecedented ability to be able to examine this invisible world. You look out your window, or you're driving in your car. You look out somewhere, you won't see any bacteria, but they're everywhere. They are the fabric of the world outside your window. They are in the air that you're breathing. They're on the surfaces you're touching. They're in your car. In fact, you have 1 trillion of them, or 100 trillion of them inside you. And so we fundamentally need to understand what they're doing and how they're responding and how they're reacting to the world around them, because that's how we're going to figure out how to make our lives better. But it's just there's so much to discover. That every day we come up with a new research project to actually explore this world. It's just a really exciting time.
I guess one of the good things too is that there's actually some money being put toward research in this field.
Yeah, I mean, people are starting to understand, especially in the medical area, looking at people's health, that the microbes in our bodies are like another organ, right? So just like your heart or your liver or your lungs, they play a role. Most of them are inside your intestine, but they're all over your skin. They're in your mouth. We find them in our lymph system. We find them, in our urogenital tracts. They are literally everywhere. And if we can just figure out how they interact with our bodies and what role that might play in our health, then it will unlock some of the great mysteries of why we get sick in many different situations. So, yeah, people have taken that on board. They now understand that, and the funding agencies and individual foundations and companies are piling in investment because this is seen as one of the areas of human health that has been ignored for such a long time. So it's now receiving the attention it's due.
Cliff, let me turn it over to you for a minute.
Well, I guess if microorganisms, make up 90% of our body, what's the extra 10%?
Yeah, well, that's a numerical count, right? So, yeah, 90% of your cells are microbial. 10% of your cells are human, but obviously the 10% of your cells that are human are very heavy and dense, like your bones, like your muscles, like your fat cells, and so those cells take up most of the weight. The bacteria in your body, the microbes in your body, the fungi and the viruses and the bacteria, they really only weigh about maybe 2, maybe 3 pounds. So that's the same weight as your brain. So if you think about, if you think your brain's important, then you might want to consider the microbiome to be important as well.
So of that 90%, what percent of the 90 is the fungi versus the bacteria? It seems like a real small piece.
Yeah, well, the bacteria take up a huge portion of that. So they're probably, somewhere in the region of, they make up about 85 to 89% of the cells in the system. And then, the fungi are there, they're hanging around. Most of the time they're sporulated. So they're living in their spores, not germinating and staying quite quiet. Most of the time, the human body has been evolved to really try and avoid fungi. Fungi are pretty nasty pathogens for us most of the time. But we do find fungi in the body which are helpful to our health. Whereas the bacteria, the vast majority, 99.9999%, are healthy for us. We need them in our body. They help train our immune system. They help our body develop properly from birth to death. And so we need them. They're vital to our health. and very few of them are actually pathogenic or dangerous to us.
And let's talk a little bit about the Home Microbiome Project. And for listeners that aren't familiar with it, maybe you could give us a little overview of what what the goals were, or what the impetus behind the Home Microbiome Project was.
Sure. So, most people would have heard of the hygiene hypothesis. This is the idea that we are now living in a world where we've taken cleanliness and sterility of our homes, of our offices, of our spaces to such a high level that we've started to reduce our exposure to biodiversity, and that may be impacting our health, leading to things like asthma and allergies and various other concerns of our modern day world. And so, with the Home Microbiome Project, what we wanted to find out was, when people moved into a home, did they bring their microbes with them and leave them in the home? Or did the microbes, including the bacteria and the fungi that were in the home, did they interact with the human? So we focused mostly on the bacteria, and we wanted to know, how many bacteria went from the human to the home, and how many bacteria went from the home to the human. But we did it in a very, very detailed way. So looking at people's noses and hands and feet, and swabbing counters and floors and light switches and doorknobs, etc., all over the homes. And we did 7 families around the US and 10 homes, and examined them daily for a long period of time, about 6 weeks. And using that information, we were able to map the routes those microbes use to move around the space. One of the key findings was that you really do just dump an awful lot of microbes when you move into a space. In your studio right now, if I swabbed the floor, or if I swabbed your microphone, I would be able to identify you as the sole occupant of that space, because your microbial signature has been deposited in that space. It's coming off your skin, it's coming out of your mouth, in your breath, and in the little droplets of water that are there. And it's just each family from our study, the Home Microbiome Project study, each family left this signature, this defined fingerprint of their own microbiome in their space. So forensically, we could identify the individual members of that family in that space, with a high level of rigor.
And did they, in return, pick up bacteria that was there previously, or did you find it was more the other way around? They contributed bacteria to the environment.
Yeah, this is a really interesting point. They did. There was some transfer from the home to the people, but it was minuscule compared to the other way. It was like firing a water pistol at someone with a water cannon. The water cannon of bacteria coming off people into that space really dominated the space. So within a few hours of moving in, it was virtually impossible to detect the previous signature of the people that were there before. Now, this is an important point. All of the people in our study were healthy, and healthy people have a rich and complex and biodiverse microbiome, right? There are lots of different species of bacteria living on them, and there's lots of those bacteria. That is one of the criteria of you being healthy. And so, when they moved into these spaces, they were epic shedders. They were just shedding bacteria everywhere, which means that, their signal was robust when it moved into the space. But if they were sick, if they had a disrupted microbiome, they'd just taken an antibiotic, or they were, they'd had some therapy or some surgery that caused the bacteria in their body to die off, or to suffer some kind of insult, then they would be far more ready to accept bacteria from their environment. And this is the whole concept of real estate, right? You have a certain amount of space inside your body for microbes to colonize, and they do a very good job at colonizing it all. But if you knock some of them back, if you open up some of the real estate in your body, then microbes from the outside who may have more nefarious reasons to colonize you, can get a foothold. And once they get a foothold, they can spread throughout your body.
And the foothold that you were evaluating, where did that seem to take place most commonly? Would it be touching things or inhaling things or drinking things or some combination?
Yeah, so it looks like the vast majority of the movement was through physical contact. But, the skin shedding, each skin cell you shed on a day-to-day basis probably has 10 to 20,000 bacterial cells associated with it, give or take. Probably give or take 10 or 20,000. But on average, you're shedding an awful lot of skin cells in an hour. So just by sitting down in a room, your standard skin cell shed is releasing lots of microbes with it. If I alcohol wipes on your hands, I'll knock back all of the environmental bacteria that have colonized your hand, but also knock back for a few minutes the microbiome on your hand, your own skin microbiome, which is uniquely yours. But within about 30 seconds to a minute, your microbiome is resurfaced on that space. So if I touch a surface now, and then I touch another surface a few seconds later, I will still be depositing, thousands and thousands of microbial cells with each interaction. And those microbial cells that are deposited, I can then swab with a little cotton swab, pick them up, extract their DNA, and identify who they are.
Now, I got so many questions. I have to figure. All right, let's go back for a minute to genomics versus metagenomics. I hear those terms and I see those terms. Can you just, for listeners, differentiate between the two?
Yeah, so if we put it in human terms, so the human genome is the genome is the DNA that you have in your cell, which is the blueprint for how your body should create itself, right? Mm-hmm. So how it builds itself. But the metagenome would be like me going into a forest and blending in a giant mega blender all of the plants and animals and everything you could see, the insects and the amoeba and the birds and everything, just putting them all in there, squidging them down, blending them up, and then extracting their DNA from that soup, and then trying desperately with some difficulty to reconstitute their genomes from this big genomic mess, right? So I've blended everything together, and then I have to try and figure out which members of that community were there. I put the DNA for the trees in one pot, the DNA for the birds in another pot, and the DNA for the tigers in another pot. But I then want to be able to infer how they're interacting. Which birds landed on which trees, which tigers ate which birds, from that mass of DNA, and that is the horror of metagenomics. But it's the only way we can really understand microbes, right? We can't see them. We can under a microscope, but they're very hard to tease apart. There is some technology being developed that enables us to tease them apart, but essentially metagenomics for microbial communities enables us to stick them all in a blender, extract out their DNA, and then start teasing apart how they interact. Who produces food that someone else is going to eat? Who kills somebody else? And those microbial relationships can really be disentangled from metagenomic data.
All right. Now, the next thing I wanted to ask is, how much do we really know about the genome and metagenomics? I mean, are we just scratching the surface here?
Well, we estimate that there's somewhere between 10 million and maybe 100 trillion, or even even more, a zillion, I don't know what comes after that, of bacterial species on this planet, right? So even with a study as rich and detailed like the home study, our ignorance is still epically profound. Even in this very, very disrupted environment of the home, the number of bacterial species we can find numbers in the hundreds of thousands. And those hundreds of thousands of species are constantly changing. They don't stay put. If I sequence the genome of one of those organisms today, tomorrow it could have acquired new bits of DNA from some of its friends. and so its genomic profile will have changed. And that really does make it very hard for us to track the genomic evolution of these organisms in these spaces. So yeah, we know a drop in the ocean, but every drop in the ocean counts when you have to deal with something as profoundly important as the microbial health of buildings and of people.
Cliff, do you have anything? I just want to make sure you get a
do.
chance to jump in. Go ahead.
And I guess because he finished up, it wouldn't be a bad time to introduce it. What I'm trying to determine is how your study of microbiome, what needs to be, or I guess what are we doing wrong in regards to infection control in healthcare settings?
Yeah, that's a very big question. The first point is to realize that if you look at things from a microbe's perspective, any built environment is a pretty bad environment. It's not a nice, warm, wet place where they can grow happily. It's a cold, dry place where it's difficult for them to grow, and that we've done that for a very deliberate reason. We've done that to try and reduce the burden of fungi and molds and pathogenic bacteria in our homes, in our offices, in our hospitals, to make sure that we stay healthy and safe. The problem is you can have too much of a good thing. Doing this, we've saved hundreds of millions of lives over the last hundreds of years across the world. Sanitation, proper treatment of air, keeping populations away from bad pathogens which can easily spread. But we now think there might be a way of folding back or rolling back some of those objectives to help us improve the microbial health of a space and potentially reduce the likelihood that children or adults will develop autoimmune conditions such as allergies and asthma, such as autism and potentially even Alzheimer's or Parkinson's. All of these conditions have a link to a disrupted microbiota at some level. And that disruption of the microbiota inside is a breakdown of the symbiotic relationships between you and your bacteria. In much the same way, the building and the building operator have a symbiotic relationship with the bacteria in the building. Sick building syndrome, conditions where we see, people with heightened susceptibility to getting sickness from VOCs and various compounds in the air. I mean, a lot of this has microbial origins and roots. But the problem is trying to figure out how to keep the right kinds of bacteria happy in this space is really, it's a tough job. We need to look at each organism and figure out what it needs, what it wants, and how to stop it from, going into banditry almost, stop it from becoming dangerous to humans. Now, most organisms, when they're out there, have no desire to, colonize human and then attack it. They are benign. They're commensal. They just like living in us, or they have particular metabolic relationships with us. But when we stress them out, when we, take away their foodstuffs, or when we cut open the gut and expose them to toxic oxygen, which they don't really like at all, or when we pump the bodies full of antibiotics, some of them can rebel. They can almost riot in the body, and when they do that, they attack the very hand that was feeding them, the human body, because all they're doing is looking for food, right? And they have automatic systems inside them. If they detect a reduction in the amount of food in their environment, they'll turn on strategies in their cell to acquire more food. I mean, it's as simple as that. And the best place to acquire food is the human body, and so you get situations where a normal bacteria or a normal fungus will become dangerous. It will become pathogenic or virulent, we would say. And once it becomes virulent, you've got a real problem on your hands. So when most people come into a hospital, they come in and they're pumped full of antibiotics. They come in and they have surgery. They come in and we physically treat the patient in a way that disrupts not just them and their cells, but also the bacteria and fungi that live inside them. We're just trying to figure out that it may be this disruption, this insult that we pay to these organisms that could have a major role to play in healthcare-associated infections. So, you know, when you go into the hospital and you have surgery and you get sick, it's all too easy to blame the doctor or the nurse or the air inside that room. We might actually need to start looking at the bacteria you brought in with you and how they were treated when they were in the hospital. And we're starting to come up with biotherapeutics, which may be able to help us keep them happy while you're undergoing your surgery. And if we can keep the bacteria happy while you're in surgery, you might be able to significantly reduce the burden of healthcare-associated infections in healthcare settings.
What's a biotherapeutic?
So a biotherapeutic is just a therapeutic, a therapy, which a compound which enables us to treat the biology of a system, right? So in this case, we've been looking at ways of providing those bacteria with the food they want at the site of surgery, so that they never detect low food and they stay happy and they don't become virulent. So we would call adding the right food in the right way in the design of that application, how we get that food, how we package it, how we deliver it to the bacteria. That's a biotherapeutic in our words.
Thank you.
So would probiotics be a biotherapy?
Well, absolutely. And if you go to any grocery store, you'll probably find a probiotic aisle. And, we find a lot of probiotics and a lot of talk about the potential health benefits of probiotics. But, that's a really enormous catch-all term that we fundamentally don't understand. Probiotics in and of themselves are just bacteria that are given to a human to have a desired health impact or health outcome. There are very, very few probiotics on the market today which have any real hard clinical data to support the claims which are made on the packaging. Some of them that we know a lot about have been incredibly useful in treating certain conditions, especially inflammatory conditions. So some of these probiotics can actually be used to treat inflammation by adding that probiotic into your diet, into your gut. You can reduce systemic inflammation in the whole of your body and treat conditions such as irritable bowel disorder. So you can imagine if we could figure out in a building environment how to add the right kinds of bacteria, you could almost come up with a building probiotic. Then adding that bacteria in to the environment, or adding the right kind of fungus or the right kind of virus into that environment, could have significant implications for improving the health and maybe even the productivity of the people that work or live in that environment. Wow.
You got me thinking. I've got a bunch of questions. We have to stop and thank our sponsors, what we call our halftime, but it's fascinating to think about adding bacteria to the environment when we've always looked at it the opposite way. And then, are we cleaning too much? Are we cleaning the wrong way? These are all great questions that we'll try and get into a little bit in the second half. We'll be back with the second half of our interview with Dr. Jack Gilbert. Great stuff. We've got to stop and thank our sponsors. And thanks to our association sponsors, the Indoor Air Quality Association, a nonprofit multidisciplinary organization dedicated to promoting the exchange of indoor environmental information through education and research. Visit them at iaqa.org.
The Restoration and Specialty Cleaners Association, who have been serving the needs of and advocating for their members for over 30 years. Remember, Triska is your link to industry training, certification, standards, and events. Their website is trsca.org. Thanks to our advertisers.
Gray Wolf Sensing Solutions, who use advanced sensor technology and embedded computers to provide superior environmental test instrumentation. Visit them at wolfsense.com.
Legends Environmental Insurance Services, the experts in insurance for environmental consultants and contractors for over 20 years. Visit them at legends-enviro.com.
And Particles Plus. They are engineers and manufacturers of feature-rich particle counters, air quality monitoring instrumentation, and vacuum pump technology. ParticlesPlus.com. Count on us.
And, of course, our marquee sponsors. John Donne Products, where restoration and abatement contractors shop. Visit them at their website, jondon.com. That's jondon.com.
Clean Facts, the #1 information source for cleaning and restoration professionals. Check them out at cleanfactswithanx.com.
IAQ.net and Healthy Indoors Magazine, a free online digital magazine for industry professionals and consumers. Subscriptions are available at IAQ.net. Please be sure to thank our sponsors for their support of IAQ Radio when you inquire about their services and products.
All right, we're back for the second half of our interview with Dr. Jack Gilbert. Just fascinating stuff. Before we broke, we talked a little bit about hand washing, and it made me just think of this question. So it sounded to me like if I wash my hands and I immediately touch a surface, I'm kind of taking away part of my defense, and I'm going to be more likely to pick up what's on that surface. Did I misunderstand what you said?
No, you've extrapolated what I said to something, another point, which is actually quite interesting. So we believe that if you have a disrupted microbiome, if you don't have a healthy, biodiverse and very abundant microbiome inside you, then you are more likely to be able to pick up bacteria from the environment. That's true. And now, the idea would be if there was a nasty pathogen in the environment and you had a disrupted microbiome, then that nasty pathogen it'd be a lot easier for it to get inside your body and do its mischief. But the problem lies in our appreciation of what is disruption. Now, there's a lot of concern over hand washing and a lot of misinformation about hand washing. A lot of this stems from the use of certain chemicals in antibacterial soaps. There was a very bad situation with a particular chemical that was used in antibiotic soaps that appeared to actually have negative impacts upon hormone levels in children. So, if they were using this antibiotic soap too much, the chemical actually altered their hormone levels inappropriately and made them unwell. So, that's something we want to avoid. But if we look at the importance of hygiene for reducing disease transmission in, say, a classroom or a workspace, then there's really nothing wrong with just using an alcohol-based solution to wash your hands. That doesn't have a big impact on your microbiome. Your hand microbiome goes very deep. Your skin is a very complex organ, and what you see on the very surface is literally just the surface. The Grand Canyon of your fingers, for example, or your hand, has an immense microbial diversity, and it goes right deep down into the skin. And so if you sterilize your hands with an alcohol wash, those bacteria come right back. I mean, they bounce back very quickly. And that bounce back means that you haven't really disrupted your microbiome. You've just made sure that if you touch somebody who was sick, or you touched a surface where there might have been a sick person interacting with it, you have killed whatever was transferred in that initial interaction. And so you've got to balance it, right? Hand washing helps. It really does help, especially with getting rid of viruses. If there's lots of sick people with lots of viruses at work, wash your hands, But it doesn't necessarily help you with anything else in the system.
So we didn't talk much about viruses. Maybe you could touch on that for just a moment. How prevalent are these viruses in our body and in our indoor environment?
Well, viruses in your body, the viruses that attack bacteria, there are probably 10 times more viruses that attack bacteria in your body than there are bacteria. So, if you've got 100 trillion bacteria, you've probably got 1,000 trillion viruses in there. So that's a lot. Viruses that make you sick, viruses that attack humans, there are many of those: rhinoviruses, cold virus, flu virus, Ebola. These are very nasty bugs, which, when they get inside you, make you sick to varying degrees. And those are easily transmitted via human-to-human contact. So if I shake someone's hand, I might pick it up. And then if I've got it on my hand and I wipe my nose, or I eat some food and I transmit it inside my body, it can potentially have a major impact on my health. That being said, it's very difficult to do that if you have a robust microflora on your hand. So there's a balance that needs to be dealt with, and it's not one that we fully understand at the moment. Our ignorance is still profound in this area. We put in measures to make sure people stay safe. So the epidemiology is a study of our ability to control the spread of disease. But we fundamentally don't understand the mechanisms by which people do get sick sometimes. We have some very good understanding in some instances and very poor understanding in others, and our knowledge is very incomplete. So if somebody has a rich microbiome, that may protect them against a virus infection. But there may be something else in their life. They may be under a lot of stress, or maybe their hormone levels are unbalanced, and therefore they are actually susceptible to the viral infection, even with a robust microbiome. So each person has a unique experience when they interact with the system. No one person is the same as any other person. Even identical twins have different microbiota. It's slightly more similar for identical twins, but overall they have a very different microbial experience. So they can respond differently to different disease states.
Now, I want to read a statement, a sentence or two from an article that you sent me. It's called Our Interface with the Built Environment, Immunity, and Indoor Microbiota. And the sentence is reducing humidity, air conditioning, and reducing the availability of complex substrates, soils, woods, through choices in building materials are likely to significantly impact microbial biomass and diversity of microbiota. This will influence the source of our body's microbiome, which could have untold consequences to our physiological, immunological, and neurological development. So I guess, are we saying there that maybe we're making our environment too sterile?
Yeah. We never make it sterile. Sterile would insinuate there's no life there at all, but we definitely reduce the number of organisms which can live in this environment. And as I said before, we've done that deliberately because, when you've got a lot of nasty things in the environment, you get a lot of human beings together and you put them in this small space, then those bugs can spread very quickly. So having that sterility, for want of a better word, has actually helped us. It's been one of our major success stories in public health. But we may have gone too far. Now, when a child is born, when the amniotic sac ruptures and the baby is born, the child, we believe, is actually quite sterile. We don't think they have many bacteria in the womb, or any bacteria in the womb. And then when they come into contact with their mother for the first time, they're colonized by the bacteria from their mother or from their father, who hugs them, or through whoever else touches them. And they get these doses of bacterial organisms, which then colonize their body, right? And then actually mother's milk has its own microbiome in it. The mother actively recruits bacteria from her gut into the breast tissue, and those bacteria are pumped into the baby. This is supposed to be. This is the way our bodies have evolved, in order to provide that baby with the right kind of microbiome to help it digest its food, its milk, but also to test its immune system and also protect it against pathogens from the environment. But then every single other interaction that child has shapes its microbiome. If it goes near animals, if it gets licked by a dog, if it grows up on a farm, if the baby is at a creche all morning, or if it's living in a 10th floor high-rise flat or an apartment, and it's not interacting with the outside world. Every single thing you do daily changes your microbial influence. And the thing about children is their microbiome doesn't stabilize until they're about 3 years old. So that 2 to 3 years of their early development, they are being bombarded by different microbes. And because their bodies are changing so much, their microbiome is changing so much, and the interaction between the microbes and their body shapes how they develop, right? So we believe that this can lead to neurological conditions, depression, anxiety, because the bacteria in our body influence our brain development. They also influence the way our brain thinks by changing or influencing nerve impulses from our gut up into our brain. And they also alter our hormone levels, and they also play roles in whether we put on weight or don't put on weight. Fundamentally, they are intricately associated with our health. So if you change, and bear in mind, we evolved to grow up in a very complex, very rich microbial experience. It was unavoidable. By removing that experience, we've done a couple of things. We've kept ourselves alive longer. We now live almost twice as long as we probably should do for our body and for our species. But we also have in our bodies all of these microbiota from the built environment. And the built environment microbiome is purely a human microbiome. So when a baby is growing up in a house, it's only really being exposed to the bacteria from the other people living in that house, right? And if they lived in houses before them and before them, as we've had for 4 or 5 generations in these kind of houses, you get a situation where the child doesn't get a rich microbial experience; it gets a human microbial experience. If you throw that kid outside and you grow up on a farm and the kid's next to you while you're milking the cows, or it's rolling around in the dust with the puppies, or it's interacting with horses, or going out and working in the fields, then its microbiome is going to be significantly more enriched than somebody growing up in a high-rise apartment. And that may be the reason why some children develop allergies and asthma, because they have a disrupted microbial flora due to a lack of microbial experience with the world. The kid growing up on the farm is much less likely to develop that microbial, the immune disorders.
Now, so you kind of answered a text question that came in, and it says, because we spend so much time inside, we're missing opportunities to develop a good biomass from outside. So if I don't know if you have any children, but if you had some young children, would you recommend making sure they get outside more often, that they get into other environments beside just being in the indoor environment they came from?
Absolutely. I live in Chicago. I have 2 small boys, they're 8 and 5. I wouldn't, I literally practice what I preach. I mean, those boys, I throw them outside whenever it's possible. Obviously, when it's freezing, it's very difficult to get them outside. But we actually brought a dog. We rescued a dog from Kentucky about 2 years ago in order to increase the amount of microbial experience these kids get exposed to. So my children now have a dog. His name is Bo. And Bo goes outside and runs around in the garden, or I take him for a run on the prairie, or he has a rich microbial experience, as you can imagine a dog does. And then he brings those outdoor bacteria, the bacteria from the soil, the plants he interacts with, the dead birds he picks up. He brings all of those bacteria back into the house. And some people might think that's a bit gross, right? But what it looks like is that increasing that microbial exposure, increasing the diversity of microbes in my home is actually probably playing a significant role in reducing the likelihood that my children will suffer from the autoimmune conditions and atopy, such as asthma and food and skin allergies. So, the dog was recruited into the family in order to play that kind of dynamic role. In Chicago, it's too hot in the summer and it's too cold in the winter. There's only a short period of time during the year when it's really amenable to going outside and playing in the woods. But during those 4 or 5 months, that's what the kids do. They go outside, they play in the woods, they muck around, they get dirty, and I fully embrace that. I even take them down to other people's farms, and I make sure they interact with the animals, like a farm party. Just keep on go and hug that sheep, go and play with that pig. Get some more microbial experience into your body.
And Cliff, I I want to make sure you get a chance to jump in if you had a question.
Actually, I'd like to go back to just some of the things that we do. In hospitals, I think that, we disinfect the rooms. I think certain types of materials are treated, to be biostatic. They add certain, chemistry that, doesn't necessarily kill things, but it tends to, prevent them from growing. I'm just wondering if you could comment on that, as well as I think that in our industry, they tend to use a lot of ATP for sampling. It was used, I think, originally in food and for food sanitation, and now they're trying to utilize this to determine how clean things are or how clean things aren't. And it would seem to me if we have this number, from the ATP, and we're not sure exactly what it is. So if you could comment on those things, I'd appreciate it.
Yeah, I mean, the lexicon of our vocabulary to describe what is clean is again woefully inappropriate potentially, or at least uninformed. We just don't understand what clean really means in terms of what we think it means. Clean, as far as we've been led to believe over the last 100 years or so of advertising, is like bleached, sterilized environments where you can be sure you could lick that and you wouldn't pick up anything. But that's not really the way in which the world works. Clean should be a healthy environment, right? And we want to balance keeping bad pathogens and dangerous bugs away from our homes and our hospitals and those environments, but also making sure that there is a rich microbial experience in that space, which, if somebody has a disrupted flora, can play a role in improving the likelihood that their health will be maintained. We've been providing probiotics to patients after surgery, which seems to significantly reduce the likelihood that they will suffer from a post-surgical infection, and also seems to reduce the incidence of post-surgical depression, so they feel better as well, afterwards. And that appears to be maybe just filling the real estate inside their body with something, anything benign, that could play a role in keeping their body happy. So the way in which we describe cleanliness in a building, even if somebody gives you a number, it just may not be appropriate. The problem is, it's all very well for me to say that, but setting an industry standard takes an awful lot of time, and it's a very complex and politically fraught process. Our best understanding is if we keep things clean, people stay alive longer. The problem is we now may need to adjust that to say, yeah, they're staying alive longer, but is their quality of life being impacted? Are we seeing people with more non-life-threatening but quality of life-threatening disorders that maybe we could treat? I'm not saying we go back to having public health nightmares of the 19th century. I'm saying we keep on mind our practices are working, but they may be hurting as well, and we need to just augment them slightly, improve them in ways in which we are only just starting to learn how to do, but that could improve it. And so number of cells in an environment, or number of colony-forming units of bacteria or fungi that I pick up from the surface, it's not a useful measure for understanding the health of that environment. It's a useful measure for determining the sterility of that environment, but even then, it's not necessarily ideal. The culture plates, those colony-forming unit plates, miss out lots of bugs which don't grow on those plates, right. But they're still alive in the environment. It's complex. It's always complex.
Let's go back to the indoor environment. Let's go back to the home. And I don't remember whether we were talking about home. I think we were talking about home. And when I read that quote about the fact that we're keeping we're using more sterile, I can't think of the right word either, products in our homes, and we need a diverse microbiome within our homes. But I don't want to I want to clarify. Are you saying we're not ready yet to add bacteria to the indoor environment? We don't know what we would add, do we?
We don't. I have some ideas, and we are constantly doing experiments right now to try and figure out if adding some of these bugs in has a beneficial effect. But we can't do that in human beings very easily. The experiment is obviously unethical. You can't just go around experimenting on people. So we use animal models, mice and rats, as a test. So we have mice and rats that are susceptible to allergies and asthma, and we're adding into their built environment using some rather novel nanotechnology solutions to embed the right kinds of bacteria in structures which those animals interact with. We are figuring out ways in which the delivery of those bacteria to those animals in the built environment could actually improve or reduce the likelihood they would suffer from a severe asthmatic attack or a food allergy. And so if it works in animals and we get approval from the FDA, we could move forward with a human trial whereby we would start designing particular, almost building probiotics to create a way to manipulate the built environment in a very sophisticated and controlled manner to elicit a specific response. The way to get around that is more the shotgun approach, right? So we bought a dog. A dog could bring a very nasty pathogen into my home somehow and make us all sick. It's just incredibly unlikely. I'm more likely to be more likely to be hit by a satellite than that to happen. But it's also highly it's a highly undirected, unmanaged way of increasing the microbial diversity in my home. And I don't know that the dog is providing the right kinds of bacteria. That my dog might not be the bringer of the good bugs. He might just bring some bugs which are helpful but not super helpful. So we need to understand that in a more sophisticated way. And the Home Microbiome Project was designed to do that. It was designed to try and figure out or should I say map the pathways by which microbes enter their room or to enter the building, and how quickly they do that. And this is a very important point, right? This is microbial cartography. We are trying to build the next generation of maps that can help us to understand how to manipulate that environment, as we've been manipulating it for the last 100 years, but manipulate that environment to benefit people's health, full stop. And we're not going to erode or wipe out, the last 100 years of sophisticated experimental design that's shown that the removal of molds and fungi, for example, have a beneficial effect. We know they do. But if we add back in some of the right ones, maybe we could have a particular impact that would be valuable for human health.
Okay. And if you could did you do any evaluation of the fungi during the home project?
We didn't in the home project. We did, but not in the published study. We know what fungi are there. There were molds, there were funguses. We know that molds and funguses have a particular role to play in managing the VOCs in the room, so the chemicals in the air, and that they can have an impact. There's a great study recently showing ergot fungus releases hallucinogenic, almost VOCs into room spaces, and it can make people believe they're seeing ghosts. Which I like that. Maybe all ghost sightings have been driven entirely by houses that have ergot fungus infestation. That's why you see it in old houses. You feel creepy, right? Because you're actually getting a slight, LSD trip off the fungus in the room that's making you see things.
I saw that article.
We know that. Go on, sorry.
I'm sorry. I saw that article and I kind of thought, wow, that's interesting, but we've got a ways to go before we prove that one, I guess.
Oh, exactly. Yeah, I mean it's fun more than anything, but it's another piece of a puzzle which maybe solved sometime in the future.
It's an interesting theory.
We look at.
It's an interesting theory, at least.
It's an interesting theory. It's a fun theory. I like it.
Yes.
But we are looking at fungi in hospitals. We're looking at fungi in cities, in urban environments, trying to understand the diversity, the ones that are good, the ones that are potentially harmful, and how we can change the built environment conditions, how we can manage those buildings to improve the outcomes of the healthy or the health-causing fungi, and knock back the outcomes of the ones that are dangerous or pathogenic, or maybe make us feel like we're seeing ghosts.
Well, we're running low on time. I have one question I have to get in. Cliff, do you have one?
Sure.
Go ahead, and then I'll get mine.
Okay. Did you study a hospital in Chicago, a new hospital? Was that your study?
Yes. So, yeah, we looked at it every single day for 365 days from 2 months before it was open to or operational to and 10 months following the start of operations.
And if you had to summarize it in a sentence or two, what did you find?
We're still exploring the data. It's an enormous data set. But one of the interesting points we found, if someone had an antibiotic while they were in hospital, they were more likely to acquire microbes from their hospital room than they were to deposit them. But if they hadn't taken an antibiotic, then that hospital room started to look like their microbiome, just the same as they did in the houses, in a matter of hours.
Interesting. And I've got a question on disaster restoration. I'm not sure if you're working with, I think it's Dr. Miller, Shelley Miller, out in University of Colorado. We've got her coming on in a few weeks, and I think she was doing some work similar to this, but before and after water damage, etc. Are you familiar with that at all?
I am indeed, and there's a number of researchers looking into this in fungi and bacteria.
And what do you think going in? I mean, obviously, I can't ask you what the results will be, but what are your thoughts going in? What interests you with respect to that topic?
So, from my perspective, it's the ability to biotechnologically design the right kinds of bugs to help reduce the instance of disease-causing fungi, disease-causing bacteria after you've had a flood in the home, for example. So, we may be able to design surfaces that are impregnated with bacteria, so that if you do have a flood, the fungi in that environment find themselves unable to compete for space. The bad organisms can't get a hold. So your home never gets into a situation where it is severely mold-laden. And we know we can do this because certain bacteria prevent certain fungi from germinating, right? And certain viruses prevent certain fungi and certain bacteria from even becoming active, and they make them dormant. And so we can figure out how to manage those kinds of dormancy or non-germinating forming bacteria and viruses, put them into surfaces, then we could prevent even a severely moist-ridden environment from becoming dangerous or toxic, and significantly improve the likelihood of a rapid and successful restoration.
I guess I should have asked this from the beginning. Now, so you impregnate this product with bacteria. Don't the bacteria need a certain environment to continue to be viable, or it doesn't matter that they're in a dry, fairly, mid-range type of temperature?
Yeah, a lot of bugs can handle extremes, right? And they enter into a phase of dormancy while they're in those extremes. The dry mid-temperature surface, for example, like on a wall, is pretty nasty for them, but they can enter dormancy during that period. And then when they interact, or when they're released, or when somebody interacts with that surface, or they live in that surface, the delivery of those bugs into the environment actually activates them. So when they get onto the person's skin or they get into a person's body, they become active. Now, if you suddenly flooded a room, then the moisture in itself would activate all of those microbes in there. The temperature gradient's not a problem. It's mostly just the lack of water.
Okay, that's great. And I know you did it. There's so many more questions, but we've got to let you go. I know you're a busy guy. We really appreciate having you join us. We've gotten all kinds of interesting comments already here. I've got a couple emails, some very interesting comments there, and people who are listening are highly respected. So we much appreciate you joining us here this week, and I hope we can get you back sometime.
Wonderful. Thank you very much. I really enjoyed it.
Thank you, Dr. Jack Gilbert. This is Radio Joe Hughes saying thanks so much to this week's guest, Dr. Jack Gilbert. That was fascinating, Cliff. Great time, and thanks for you for helping out.
Fantastic.
And of course, to my engineer, John, you got to have faith. Our growing group of loyal listeners. And Cliff, did you have a final word?
No, I didn't. I'm good.
Very good. All right. We'll be back. Oh, by the way, next week we're going to do a little discussion we've got Maury Astley and I think Brian O'Halleck's coming on, and we may have a few other people coming on. We're getting into the disaster restoration conference season, and it's time to talk to those folks, see how things are coming along with the new Council of Associations. And then Cliff and I will be hopefully joining you in a couple, maybe 3 weeks from the RIA conference. So we've got a lot of great stuff planned. As I mentioned, we've got Dr. Shelley Miller coming in May, and we're going to continue doing our best to get these great researchers and thinkers on the show as often as possible. This was a lot of fun. So everybody, please come back and join us. Next Friday, we'll be back. That's my birthday, April 17th at noon, with the next broadcast of IAQ Radio.
This has been another IAQ Radio production.